Hello, everyone. So in this lecture, we're going to talk about our next biomolecule that is important for us to know a little bit about, and that is the lipids. So let's get started. Um, by the end of this module, you should be able to identify the building blocks of lipids, explain the structural and functional characteristics of lipids, and summarize how saturation affects a lipid's characteristics. So, um, lipids include several different types. We have fats, oils, waxes, and steroids. Um, and these are all organic molecules, just like carbohydrates are organic molecules. So they're going to have carbon, hydrogen, and oxygen. Um, in general, lipids are hydrophobic. And so hydrophobic means basically doesn't like water, right? There are modifications that can be made to these lipids, including the ability to be amphipathic, which will, or amphipathic, which we'll talk about here in a few. And there can be really several different forms of lipids. So we'll talk about some of the different types that you can come across. And so first we need to get a little bit of basics on lipid structure. We have fatty acids that can be saturated or unsaturated. So when we're talking about saturated fatty acids, we're talking about this chain here, right, of all these C's. And if they're saturated, that means that there are no double bonds, right? So all of these C's have single bonds. Carbon has the ability to bond to 4 different things. So you see that there are 4 different bonds coming off of each of these carbons in this chain. And therefore, we call that a saturated fatty acid. Now, don't get confused by the double bond up here. That's part of glycerol, so that's not included in the fatty acid. Now, these fatty acids are going to be packed really tightly together. They get pretty linear chains, so there's no real space between them. And therefore, you get, they usually come in like a solid form at room temperature, right? So this is your butters, your lards, other fats along those lines. Now, our unsaturated fatty acids are going to be unsaturated. So that means that they have some double bond. So if we look here, we see a double bond introduced into this fatty acid chain. And therefore, we have a monounsaturated fat, right? A single double bond here. Now, when you induce these double bonds, you get kinks in the chain, right? They no longer are linear, they kink. And therefore, this is going to actually prevent it from tightly packing together. And this is going to be like your oils, your corn oil and your olive oil, stuff like that, that would be not solid at room temperature. Now, your saturated fats and unsaturated fats come in a couple of options. So your butyric acid, saturated fat. Here, you see no double bonds, so it's saturated. Our monounsaturated fats are going to have a single double bond, and then our polyunsaturated fats are going to have 2 or more double bonds, right? Now, when you form these fats, you're going to get them by combining a glycerol, which is this guy here, with some sort of fatty acid through a process of esterification. Okay. And so you can get fatty acids in mono, di, or triglyceride format. Here we have a triglyceride, so we have our glycerol and then 3 fatty acids coming off of it. And so just keep that in mind. You have all 3 options for your fatty acids, and the process of forming those mono, di, or triglycerides is through the process of esterification. We can also have a type of lipid called a wax. This, for example, is beeswax. You get this fatty acid connected to this long alcohol chain, all right? And so this long chain alcohol takes the place of glycerol, and that's basically what's going to give you your wax substances. Then you have these kind of specialized versions. You have steroids and sterols, which are both lipid-derived compounds. And your steroids, you have your 4 hydrocarbon ring structures. So 1, 2, 3, 4. In this case, we have testosterone. And then in our sterols, we get some modifications. We get this functional group added down here, and then we get this alcohol group added down here. And this is cholesterol, which is a fairly common sterol. And so here's kind of a summary of those different types. We have our fats and oils, which are your glycerol connected to your fatty acids. Then we've got our waxes, which is our long chain alcohol connected to our fatty acid. And then we have our 4 hydrocarbon ring for our steroids, and then our modified steroid to form our sterols. And remember that OL, like in glycerol and in cholesterol, that suggests that there's an OH group. It's an alcohol group. And so that's what that OL suffix means. Lipids have several different functions, one of which is energy sources. So your body or microbe can break down lipids for energy. They can be used as part of the cell structure, and they can also mediate cell signaling. So let's look at some of those special scenarios. So here are phospholipids. They are a common part of cell membranes. They are amphipathic. And what that means for amphipathic is that you have a hydrophobic end and a hydrophilic end, right? And so this side likes water and this side doesn't like water. And then you get this kind of orientation where you get these lipid bilayers, right? So you have one layer of phospholipids here, one layer of phospholipids there. Excuse me. And the hydrophilic end is going to face outwards. And so that's going to face towards where water will be. And the hydrophobic ends will face towards each other where there is no water. So we have glycolipids. So glycolipids are a lipid linked to a carbohydrate, right? So you've got your lipids down here, and then you've got your sugar residues, and this is the glyco part that makes the glycolipid. So you get these on the cell membranes. These can be used for cell recognition. They can help maintain cell wall structure. And so they're common on pretty much all organisms that I'm aware of. We have lipoproteins. Lipoproteins are lipids linked to a protein, right? And so these can be put into cell membranes to help stabilize those membranes. This is just an example of what you might see. So we've got these different phospholipids here, and then we've got these ApoA and ApoB, and altogether this whole thing makes a specific lipoprotein. We have LPS, or lipopolysaccharides. So based on what those words mean, lipo, that's the lipid, and then the polysaccharide, we should know from the carbohydrates lecture, is multiple sugar molecules together. And so this shows you how the LPS can come off of your cell membrane. This is the toxic part of the gram-negative bacterial cell. So you'll find these on gram negatives. And this, when it gets released into the body, it can cause a toxic effect to the individual who's infected with this organism. We've got mycolic acid, which is this guy up here. So mycolic acid is this special part of the bacterial cell wall for mycobacteria. The big famous mycobacteria are tuberculosis and leprae. So these cause TB and leprosy. These organisms are acid fast organisms, right? So we don't talk a lot about acid fast in this class. but they're not gram positive or gram negative, which we'll talk about later. And we'll do some of that in lab, but they are acid fast. And these mycolic acids are what lead to that. And they are part of the virulence factors that these organisms have when they're causing infection. And then when we have things like cholesterol, these are like cell membrane molecules. Uh, they are not common they're not very common in microbes, but they are very common in animals. So they can provide some rigidity to membranes and fluidity. All right, so with that, you should be able to identify the building blocks of lipids. This glycosidic bonds one got left in, so I apologize for that. You should be able to explain the structural and functional characteristics of lipids and summarize how saturation affects a lipid's characteristics. With that, there should be a objective check in D2L, so give that a whirl and see how you're doing on these objectives, and have a great day.